Hi, I'm Vamsi Chodasetti. In meiosis, there are 12 steps for the cell to divide into 4 daughter cells. The first step of meiosis begins with the interphase, in which chromosomes are duplicated and each replicated chromosome consists of 2 identical chromatids. These chromatids are joined together at the centromere. The second step of meiosis is prophase 1, where each replicated chromosome pairs with its corresponding homologous chromosome to form a tetrad, in the process called synapsis. A tetrad is a pair of homologs that has 4 chromatids, 2 for each chromosome. As a homologous chromosomes form a tetrad, their maternal and paternal chromosomes cross over one another, swapping genetic material. These points at which crossing over occur are called chiasmata. Crossing over produces new combinations and variations in the genetic code called alleles. The nuclear envelope dissolves and the spindle forms. The third step of meiosis is metaphase 1. The tetrads line up on the metaphase plate at the cell's equator, and the tetrads line up randomly on either side of the equator. The spindles attach to each tetrad. The next step of meiosis is anaphase 1, in which the spindle fibers contract and pull each homologous chromosome pair towards the poles of the cell. In the fifth step of meiosis, telophase 1, the chromosomes untangle, the spindle dissolves, and the nuclear envelope reforms. In the final step of meiosis 1, cytokinesis occurs. In cytokinesis, 2 cells are formed. Each of these cells are haploid, having a half set of chromosomes. Because of crossing over and random assortment, they are genetically unique. After cytokinesis is over in meiosis 1, interkinesis begins. The difference between interkinesis and interphase is the chromosomes are not duplicated. After interkinesis, prophase 2 occurs. The chromosomes are condensed, the spindle forms, and the nuclear envelope dissolves. During metaphase 2, the chromosomes line up on the metaphase plate and the spindle attaches to the centromere of each chromosome because they are no longer in tetrads. In anaphase 2, the spindle contracts, the centromere split, and each sister chromatid is pulled to the opposite pole. In the final steps of meiosis, telophase 2 and cytokinesis, the divisions of the nucleus and cytoplasm occur. Each daughter cell has produced one more daughter cell, so that, in all, meiosis has produced 4 haploid daughter cells, each genetically unique.